video. We're continuing on. I'm studying this chapter, making these videos for my AP Bio students so that they don't have to read the textbook or study over a break if they don't want to, but that we're fully prepared to jump into our next unit. So hopefully these videos are helpful. If they are, definitely let me know. I appreciate your feedback. Okay, so we're on to chapter 14, talking about Gregor Mender, my favorite biologist ever. Yeah, that's true. As it is approaching Christmas, I just want to just mention I had a great time last night studying and being reminded about the reason for the season. So I just want to mention I do all that I do as a teacher because of that, and so I hope that I can be more and more like the great teacher with a capital T every day. So that's all I'll say about that. Let's jump into the chapter. But yeah, I just wanted to mention that as we do get to celebrate Christmas this week, and that's a very meaningful thing to me. Okay, so Gregor Mender, who was also a Christian, he was in fact a priest. And this is actually not just a fun fact, this is actually very relevant to his work. And the reason why is Gregor Mender studied pea plants. And the laws that he was able to determine and figure out about genetics without even knowing the existence of DNA or chromosomes or anything else that we do today, he was able to do his work because he was an amazing scientist, first and foremost. He was a Christian who was trying to understand the way that God made the world, secondly. And thirdly, because of the incredible fact that he studied pea plants, which are very simple genetically speaking. In fact, he was originally trying to answer certain questions about the genetics of humans. But if he had studied humans, or even if he had studied lab rats, which he wanted to do, they don't mention it in the textbook, but I know from my own studies in his life that he actually wanted to study rats. Because, rats had been commonly studied to better understand genetics in humans, even at that time, presumably. And he wanted to study rats, but then his boss, I forget what you call the boss of a priest, maybe a bishop, maybe the local bishop told him no, he could only work with plants for his studies because it was unethical for him to work on mice or animals. So for all those reasons, and this directing of life, and even him getting turned down for certain opportunities, he ended up spending a lot of time studying pea plants. And that actually was perfect because pea plants are simple enough for him to have drawn really amazing conclusions and really give us a level 1 understanding of genetics that we would not have without his work. So all that to say, let's jump into his life and work. So Gregor Mender was a priest / scientist. There wasn't really a lot of professional scientists back then, especially in the field of biology. So he was a priest who was studying the characteristics of garden pea plants. He used a scientific approach to identify actually 4 laws of inheritance. The 2 main ones that you need to know for the AP test we emphasize are the laws of segregation and the laws of independent assessment. He had actually 4, which I'll mention today. Okay, so the book says he chose to work with peas. He was actually really incredibly lucky and blessed to work with peas. He actually, as I mentioned, didn't want to work with peas originally, but he ended up working with peas and ended up liking it, and it ended up working out very, very well for him. Once he decided to work for a plant, he just chose peas. That was a brilliant choice. He would not have been able to perform his experiments without peas, without it being peas. And he did several important experimental design decisions that involved making sure that led to him being able to completely control which plants bred with each other, which ends up being very important. he would study the character or the heritable features of the plants, and of those characters or traits, which is the variation of characters, he found actually through his work over many years, he found 6 traits that followed certain rules, which end up being called Mendelian genetics. Okay, so let's jump to the main idea. We'll jump through the vocab. So here's the main idea. Is right here, illustrated by the color of the plant. His main idea was this, and this is what he discovered. When he crossbred 2 plants, one of which had purple flowers, one of which had white flowers. He did this for 5 other traits as well, but this is a nice one to look at to start. So he bred purebred purple flowers and purebred white flowers. What does that mean? Well, we'll get into that in a second. But what he found was the next generation was 100% purple. Wow, that's surprising. Alternative hypothesis, or what people thought about genetics at the time, is that it would just be a blend of the 2 parents. So they thought all the flowers would be pink. Why do people think that? Well, that actually does happen in real life in certain plants in certain situations, but not in Mendelian genetics. Okay, so that's the first surprise. Everything was purple. Well, what happened to the white plants? Is that DNA, to use our modern understanding, is that DNA just gone? Did it not get passed on? No. What Mendel realized, which was brilliant, was that the white trait was not gone, because he took it a step farther than people had before, and he raised a new generation, and he found that about 1/4 of those were white. Why was this? Was it a genetic mutation? Was it something strange going on? No, actually, it was really simple. It seems simple to us, but it was a hard thing to really understand without understanding genetics. But what he realized is that the white color trait was a recessive trait. It was something that could be passed on without being expressed in the phenotype. So what does that mean? Well, the genotype is the actual genes. It's what's in the actual DNA for the plant. This has a genotype of 2 alleles telling the flower to be purple. And this has the genotype of 2 alleles telling the flower to be white. And so the next generation, to use a Punnett square now, would be this. So we'll use a capital C to represent the dominant trait. This is the convention of how we do Punnett squares. We'll make it a little bit more obvious that the capital C's. We have a lowercase c for the white flower because that's a recessive trait. Now, according to his conclusion, which this is called the Law of Segregation, 50% of the time, each allele gets passed on. Now, in this case, it doesn't matter. Even though it's 50% this allele, 50% this allele, they're both capital C. They're both that dominant purple color. So it's always passing on to its offspring, like that trait. So 50% it'll be this allele, 50% it'll be that allele, but they both happen to be capital C. And the same for the other one. So it's 50-50, but they're always passing it on. Well, what's happening here? Well, all of the next generation, the F1, or the generation after the parental generation, the first generation, all of them are what we call heterozygous. Well, what does that mean? Well, it means that they have both traits. They have the allele, or the genetic information found on the chromosome for both the purple trait, the dominant trait, and the recessive trait. So what happens? They get the dominant trait. That's why it's called dominant. But what happens in the next generation? Well, once this generation was crossed, now, if we did another Punnett square, we'd see that just by the random segregation, we would start to see that white generation pop up again, because using another Punnett square, we can see that 25% of that would result in the lowercase c, this characteristic from the P generation has now come back in that recessive trait. Very interesting. Let me just correct a couple common misunderstandings at this point. Misunderstanding 1, This is not a genetic mutation causing the next generation to be white. No, it's the trait, the alleles, the genetic information was always there for that, but it was just covered up in this generation. Notice all four of the possible, all four of these boxes, which these represent the possible offspring, are capital C, lowercase c, meaning all of this generation has the genetic information for the white flowers, but they also all have the genetic information for the purple flowers. They all end up being purple. Again, this is not a genetic mutation. This is just showing why the first generation was all purple, and then in the next generation, the white showed up again. Very important. Second misconception is this, that I often hear from students, is they get confused and they think that, I forget what I was going to say. Oh, wait. Oh, they just forget that this is all based off of meiosis and about the genes. That this doesn't mean a flower is 25% this, 25% this. They just get confused in the math and the probability. This is just saying each individual flower has a 25% chance of each of these traits. And so it doesn't mean that, so it just means if you grab a random flower from the F2 generation, you have a 25% chance of getting a white one. If we make a new flower in that generation, it's a 25% chance of being a white one. It doesn't actually tell us anything about individual, telling us in the whole population, about 25% will be this, about 75% will be that. And in fact, again, what confused people is in real life, it's not going to be exactly 25, 75. Mendel experiments weren't exactly that, but he got so close to it that he hypothesized that it would be about, that this would be the statistical average. This would be the resulting generation would be about 3 to 1. And he found that for 6 different traits. That's really incredible. So what should you take away? So Mendel did work on pea plants. It was really impressive work that could have only been done on pea plants because human genetics, as we'll find out in a future video, is much more complicated. But through luck and through being blessed, he ended up working with pea plants which demonstrate this type of dominant recessive Mendelian inheritance for 6 traits. Yeah, and so it ended up working out. He came up with 4 laws. I'll just read them to summarize them for you real quick. Number 1 is he was saying that there's alternative versions of a gene that can give contrasting instructions. So we had the purple flowers, we have the white flowers. We'll see in another slide, there's a flower for a wrinkled seed pod and there's a smooth seed pod. There's these contrary instructions. With pea plants, just only is it binary, which was nice. It was either purple or white. We don't have a large variety of colors like we see in other flowers, for example. And then for each trait, they have 2 genes, 1 from each parent. He also was able to show how it's being passed down from each parent. And again, that was because of his experimental design. And then the law of segregation, in which he said each parent is passing down only 50% of the genetic information to each one. So it's a 50-50 chance whether it'll pass down in the heterozygous case, whether it will pass down that purple trait, whether it would pass down that white trait. It would pass it down to about 50%, each one to about 50% of the generation. And then he coined the terms dominant and recessive trait to explain why purple is showing up more than white. So that's the basics. I hope that was helpful. And that's all I have for you today. Okay, so that's your quick introduction to Mendelian genetics. Have a great day.